Hey.
Hello, how are you? Hi.
Hi there.
Hi, how are you?
Good. Feeling unprepared for the paper, but
Oh, yeah.
but I have a window of time.
Oh, yeah.
I'd love to hear the discussion.
Yeah, that would be great. Thank you. Nice to see you. Amanda, are you okay?
Can you hear me?
Yeah.
Okay, great.
Right. Will Jesse be joining us, or?
To my knowledge, yeah.
Okay.
It's nighttime there.
Well, yeah, it is already dark.
Yeah. Now that it's daylight savings time has ended, the sun sets at 5:30 here.
So, yeah. So, did you want to start, Amanda, or do you want to wait for Jesse?
Sure. I guess I'm not prepared to like share my screen and do all of that. We can start with like general thoughts about
All right.
the paper. So I'll start with, I guess, sharing, and I shared a little bit in the Slack about why I thought that this paper was interesting and why. So I was saying the reason why I wanted to read this paper was Weiner at one point. I wrote down the pages. From about page 55 to page 57 of Human Use of Human Beings, he talks about how in insects, like there's no real sophisticated memory system. And he says that through the process of metamorphosis, there's no way that memory could remain. He says the physiological condition for memory and hence for learning seems to be a certain continuity of organization, which allows the alterations produced by outer sense impressions to be retained as more or less permanent changes of structure or function. This is Weiner. Metamorphosis is too radical to leave much lasting record of these changes. It is indeed hard to conceive of a memory of any precision which can survive this process of radical internal reconstruction. But then this Levin paper is a review of research that basically says otherwise, specifically regarding metamorphosis, but also regarding just like other instances of structural brain changes. So one example he talks about, the Levin et al. Talk about, is squirrels when they hibernate and their brains experience like dramatic cell death, but they recover very quickly. And there are some things that they can like learn and remember, and then some things that they cannot. So, well, the first part of what Weiner says there, that memory and learning seem to be like changes in organization, like that seems right. But I guess what this research shows is that the structural changes associated with memory are not just like physical. Like the synaptic plasticity hypothesis is the main thing that they're kind of arguing against. It's not just changes in brain chemistry and neural connections. So I guess my impression after reading this was that there's still something really compelling about Weiner's take that there has to be some kind of continuity of organization. Like in a sense, that's what it means for an organism to be the same organism it was before. To like for a life to have continued, there has to be some continuity of organization. So yeah, but this research makes that more complicated in ways that I like don't fully understand. And a lot of the paper was gesturing at like where future research needs to go. Um, I kind of saw the paper as pointing to anomalies for the synaptic plasticity hypothesis, um, both anomalies that occur like in cases of the brain, but also in non-neural kinds of memories.
Okay.
Huh.
Yeah, that's great. So I don't know. I guess Jesse's here. Hello, Jesse. I don't know if he
No.
wants to share his screen and everything, or because we're kind of
Yeah, it'll take me a few minutes to share my screen, but I can do it. But if
Yeah.
anyone else wants to, they can too. It's been like a few minutes.
all right. So yeah, I don't exactly know which one. Let me share my screen. In the meantime, we can go over like what paper these are and what we're talking about. So I have up the Notion page. So this is the Notion page. So this is, of course, the Blackiston et al. Paper. Is what you're referring to here, or is that the other?
Yep.
Okay.
No, Levin is one of the three authors.
Okay.
Watson's the first author, and then those quotes are I just read the highlighted bit of the first one.
Okay. So yeah, this is from Human Use of Human Beings. Yeah. And put that in. And then this is anomalies for the synaptic plasticity hypothesis. Okay. And so that's where we are with that. And basically, they're challenging this idea of the synaptic plasticity hypothesis. So this is our document, the stability of memories during brain remodeling, a perspective. This was actually from 2015, and this is from the Communication and Integrative Biology Journal. So, and yeah, this is where they ask the question, could stable memories persist when cellular turnover and spatial rearrangement modify the hardware within which experiences are stored? So in other words, if you had a brain that was like basically turned over, I guess in the case of the squirrel, and there were significant rearrangements, could you remember things? Could you have stable memories? And they say yes and no. So they have some examples in here where they kind of talk about both sides of that. And then, so they actually refer to the engram, which of course is this old idea that there's a physical location for memories. And we've talked about this in the Saturday meetings. We've talked about like the molecular basis for memory. But the engram hypothesis was, they basically argued that there was a certain place where memory was stored. It wasn't just like synapses. It was, maybe cells, maybe something else. And, not necessarily the idea of localization of function, but that there was a place where you could delete or retrieve a certain memory from. So that 16 and 17 are the references there. And then the question that kind of follows from that is, how is information encoded in living tissue? And what is the relationship between memory and the spatial location of specific physical modifications? So it's that spatial location in the brain. And you have cells and you have synapses. We know that like if you interfere with a synapse using a pharmacological agent, that you can sometimes reverse memories. And like they've done this in rats. But if you take like large chunks of the brain and remove them, actually we know from the patient HM that we can also induce amnesia through doing that. So I mean we know from like just parts of the medial temporal lobe, if they're removed, you can remove the ability to recall memories or encode them. So there are different types of amnesia. So that's known. But the engram hypothesis, I don't know what the status of that is. I think it's kind of in disrepute. They never really found the engram, but this is an idea Let's see, of 16 and 17. Let me zoom
Yeah,
in
a
here.
lot of interesting looking citations.
Yeah. Oh, yeah. We
Just a
have
lot,
over
period.
100. Yeah. So citation 16 is mammalian brain substrates of averse classical conditioning. This is from 1993. This kind of talks about the engram. 17 is 50 years since Lashley's In Search of the Engram, Refutations and Conjecture. So this was from 2001. So this was back in the early 1950s when they talked about um, the Lashley's hypothesis about the engram, or at least his paper In Search of the Engram. So this is an idea that's been around for a while. So yeah, there are a lot of things in this I wasn't, I'm probably not aware of myself. A lot of this one, information in the brain, a molecular perspective. That we've talked about, but just kind of like the latest on the engram, I'm not really sure of where that is. But I see that they have, of course, Levin. He has done a lot of work with planarians or the flatworm, where they can actually, flatworms have a great regeneration capacity. So you can actually take a single cell of a flatworm. You can like just basically blow away everything but a single cell, and you can get the flatworm to remodel its entire body, and it can even remember things from the previous form. And
Right. One of the experiments that they talked about is they cut planaria in half, and the side with the head retained the memories, but also the side without the head, once it regrew, it still had. Um, it wasn't as strong of a result. Um, I think it said that they learned the task more quickly. Um, so it's not like they fully remembered the task, but they showed an increased capacity to relearn it.
yeah, so then there's this. So actually, they show a number of different examples here where you have holometabolous insects, which are the insects that undergo metamorphosis, and these are caterpillars turning into butterflies. It's the most obvious example where you're pruning in many neurons to the cell body before generation of adult-specific brain regions and sensory structures. So you have that. You have planarian flatworms where there's remodeling, regeneration of partial full brain from the remaining tail fragments. So that's the example Amanda was just talking about. Behavior surviving brain remodeling. So in the case of insects and flatworms, we have these differences. So the butterfly, or when you get a butterfly, you retain some behaviors but not others. When you get a flatworm, you retain a lot of memories, classical conditioning, for example, and you also get this more rapid learning and recall of environment. And then you have Arctic ground squirrels, which do hibernation. And there's a remodeling event there where they have extensive pruning of their dendritic trees of the brain. So this is where the dendritic trees prune away, and this is typical for like high stress environments where if you raise a rat without a lot of environmental stimuli, like if they're raised in an empty cage, or sometimes if a small mammal undergoes starvation, they'll have the same problem where you get pruning of dendritic trees. In this case, they're going into hibernation, so their dendritic trees reduce in size and number, and that morphology is restored. And so you get a lot of behaviors that are retained during or after the remodeling, but not all. We don't. I mean, I guess you could. There are a lot of things that are retained, but they don't remember everything. This is interesting. They didn't put C. elegans here. C. elegans dower stage, which is where the larval stages of C. elegans, you get this stage where it's sort of a high stress, high starvation stage where the worm, grows a thick cuticle, and there are a number of neural changes, and they recover from the dower stage. But there's some indications that there are differences in worms that undergo Dower, their Dower phenotypes, for example. And so there's an interesting literature there as well. So these are examples of this. I mean, I don't want to go through a lot more of the paper without giving people a chance to weigh in, but just here's another bit about the engram here. So, maybe we'll talk about that a little bit, or maybe not, but basically there's this theory of the engram, which is a physical chemical memory trace, which could perhaps be transferred into an untrained animal. And so people have been searching for the engram as a discrete physical unit in space that they could maybe transfer to different organisms. But so that's a little bit background on that.
I had a question about that particular paragraph. After the highlighted bit, it says animals that were fed on the worms that were fed on trained worms, like they've trained the worms and then fed those worms to other worms. Is that what that is saying?
Yeah, I think so. They were transferring the mRNA in the brain, so they were eating that. And so, I mean, so they will use planarian extract. So they'll like grind up a planarian and they'll feed it to other planarians. I know it's cannibalistic, but I don't think it's a big deal in planaria. But
that part was just really interesting.
Yeah, so there's this issue with RNA. So basically, they're transferring RNA doing that. They'll just grind it up, and the tissue still has the RNA in it. But planarians aren't that big, so it's basically just the cell extract. And you see cell extract like in cell biology. They'll use it for different things. People will try experiments. We'll have like cell extract that is uptaken by some other cells, and that people will make claims about it. This is where they're transferring the RNA basically to other worms, and it's this experiment in '86 that where the RNA was the active agent allowing memory transfer. So you can either expose them to the RNA or not. So this is where they're uptaking it from the environment. There are other naive worms injected with the RNA extract from trained worms, demonstrating the saving effect. Well, RNase, which is an enzyme that removes RNA, this particular RNA, as well as others in the organism. So if they do an RNase treatment, it abolishes the effect. So they could basically pinpoint the effect down to the RNA. They could try putting it in the food, in the environment as food, or they could put it as extract and inject it directly into the worms, or they could abolish the RNA through using RNase. And so this is they didn't know much about at the time of these experiments how RNA worked in terms of the brain, its different regulatory functions, but this was all kind of this set of experiments here. So this was James McConnell and Robert Thompson, So while basal meaning that they're not, that they're I have this problem with lower or higher organisms, but basal just means like it's not as, not as derived, I guess, in evolution and phylogeny as, say, like mammals. So it's like closer to the base of the tree of life. I guess you could say that, but anyways. Planarians possess a well-differentiated nervous system and central nervous system with chemical synapses. That means that, planarians, I guess the implication of basal meaning it's not as, their nervous systems aren't as highly evolved, although, I don't know if that's true or not, but, have as much complexity, I guess, is a better way to say it. Planarians possess a well-differentiated nervous system and central nervous system with chemical synapses. So chemical synapses, of course, you see this. You even see them in sponges, but they're not contained in the nervous system. But in planarians, in C. elegans, you have these nice nervous systems with a central nervous system with cells and chemical synapses. And this is what we need for synaptic plasticity. So we can do these kind of experiments in these very simple organisms. You know, they don't have that many cells. They don't have a long lifespan, so we can study like, you know, inheritance. We can study, you know, we can trace things down to the single cell, and so this is something that they've been looking for since Hebb proposed his model of memory. So this is where Hebb proposed synaptic plasticity as a mechanism for memory encoding, and they needed to test it somehow in a very definitive way. So they were doing this. They were working with early experiments with planarian, and this is what they one of the experiments that they tried. So this is great stuff. Morgan, how are you? Can you hear us or?
Oh yeah, yeah. No,
Okay.
I can you hear me now?
Yes.
Good. Yeah, no, sorry, I had problems, but I got the headset now. Yeah, I mean, I'm familiar with some of these old planarian experiments, and you know, the references are actually history of neurosciences papers, not the original, not until you get to the later 66, 67 work. But wanted to go back to something you said about rats in deprived environments going through that same kind of pruning, and you know, I'm certainly familiar with some of those experiments, and not familiar with the particular Arctic squirrel.
Yeah.
And they're coming out of torpor and then having a kind of regrowth, right? You know, I'm curious if anybody's looked at what are, you know, the contrary, you know, interventions that bring back some sort of equivalent, you know, regrowth or reconnectivity in deprived. I mean, you know, like again, put those same rats now into you know, a different environment, a rich environment. You know, will they? You know, how? You know, critical periods. Will there be some growth? You know, what can you do to intervene and you know, restore what we might call normal function, or something back to equivalent.
Yeah, that's a good question. There, like, you mean like a training regimen that you might give a squirrel to?
Perhaps. Yeah. I mean, I certainly, you know, certainly, like in the history of neuroscience, we've done a lot of deprivation experiments,
Right,
a lot of like, this is how we destroy function. You know, I'm surprised, or you know, like it's interesting. It's yeah, I wouldn't have guessed the squirrel case.
Yeah.
Like I didn't realize that there was both a pruning and then a regrowth when you're coming out of the torpor.
Right, because it seems like as soon as the squirrel gets just like back into its normal environment, starts, you know, doing squirrel things, that's all it takes. It's not like no like intervention is needed in this case. So
yeah. Or it's certainly part of the physiological process. Like they know this is going on, and they know it needs to be restored. Right. Again, like I wasn't quite sure. It just says restoration of morphology shortly upon arousal. Again, I'm sorry for just skimming paper, but I just wanted to join in. And but super interesting.
Yeah, I imagine it's kind of like when you if a human goes into space and their muscles atrophy and they have to train them to get back into shape, or if you go into a long-term recovery from surgery. You know, it's like you get this this host of physiological changes that are associated with the sort of torpor, and then they get into their regular lives and they train back up. But I think the other
Mm.
question you're asking is that
Mm.
how could you design an intervention for a squirrel so that
Mhm.
they get like super performance or that they can recover above baseline?
Or certainly restore a baseline, right?
Yeah.
Yeah, or something equivalent. Yeah.
So there've been some experiments where they've kind of wondered about that and tried to think about ways to like enhance that kind of adaptive uh, refractory period. So I'm familiar with well some of the work that they've done with motor switching, where you like put a weight on you put a weight on your arm or something when you're learning some task, and then you take the weight away and you can enhance memory, or you can interfere with memory selectively. There's also some work that I remember reading like a long time ago. It's like the Japanese developed this katsu technique, and the thing is like where you take like a tourniquet and you put it on someone's leg or arm, and then you have them like lift weights, and then you take the tourniquet off, and like there's this rush of blood through the system, and then you continue to train them on like lifting weights or doing something, and like the reperfusion of blood will like the tissues will respond at like 2 or 3 times the rate of like what you would expect normally from the training regimen. So it's like you're doing your what they call preconditioning the tissue, and this happens not just physiologically but from like a procedural learning standpoint as well. So there are different interventions you can use for that. It's kind of interesting work that it kind of comes out of like training astronauts so that you minimize the amount of muscle mass that they lose when they go into space and things like that. So that's one thing I'm aware of in that area. um, yeah. So
I mean, it's a. I mean, it's definitely a good example. And but even to some degree, I would almost call that simpler. Or like uh, uh, again, like restoring nervous system function has historically been a harder task,
right.
and it's good to point out that astronauts suffer long-term consequences of their trips.
Right.
Like we don't restore them to normal function. We just get them towards in the right direction. Um, so I mean, like you said, it's an active area where it's they've because isn't the senator who's asked, doesn't he have a twin?
Mark Kelly. Yeah, he does.
The one like there's an astronaut that has a twin,
right.
And so they could really compare them in terms of the one who's gone up and the one who hasn't.
Right. Yeah.
But they've gone through all the same training and things like that.
Right. So Amanda, did you want to say something?
Well, one thing that would be interesting to look into, because in the squirrel example, hibernation is this dramatic like decrease in metabolic rate for a sustained period of time. It's like the organism's functions just like across the board like slow down. So if we were trying to think of what types of interventions could improve the regrowth or prevent like cell death or something, prevent like neurodegeneration, for example, then maybe metabolism related things would be an interesting place to look. If it's this slowed metabolism and torpor that that causes the dendritic shortening or whatever, and then it's the recovery from that that's associated with the recovery of cognitive function, then maybe and this has come up in other things that we've read, and just like made contact with as a group, the link between metabolism and cognition. So yeah, that could be an interesting place to look for possible regenerative like interventions or mechanisms.
Yeah.
Yeah.
Go ahead.
I agree with that, and also like apologies. I had to kind of handle a bunch of stuff. I'm actually still running around. There's a few questions I'll try to bring up later, but really happy to have you here, Morgan. I'm glad that you could share your insights and be part of this. So I want to say that for sure.
Sorry, my son just called and he needs me to pick him up.
Okay.
It's okay. I'm glad you're here.
Sorry.
It was worth it.
Apologies, and I will catch it later.
Okay.
Yeah.
Well,
Good to
thanks
see
for
you.
attending. Yeah, good to see you.
Okay.
Great, thank you.
All right, thanks.
So yeah, there were a few things that I wanted to mention earlier, and I know you kind of went through the paper, and I didn't hear all of it, so I'm going to kind of circle back around that. But definitely, from what Amanda was saying, I think you can hear me okay, right?
Oh
I
yeah.
think I'm okay. It's cold outside and it's noisy, but like I'm trusting that this is working right. So Amanda was talking about metabolism, and some of the questions I was going to bring up, and I think we covered in earlier, had to do with sort of these other not mitigating factors, but these other things that are happening along with the main thing. Kind of like what is linked or what is going on with these processes. In this case, memory, but like it's a little bit related to my Cognition Futures methods project comments about tracking across domains. And what comes to my mind about metabolism is sort of like metabolism isn't just it's not really a location or an organ. It's kind of a systemic moving of a lot of parts. And I find that to be interesting in light of what we're talking about, because I wonder how some of that I wonder how that general like to kind of circle back into some of the questions I had earlier. Like there were some things along the lines of somewhere in the paper there's discussion about I don't think this is a major point, but somewhere in the paper I think it was like a minor thing about describing like the conditions of one of the experiments, but they noted like something about the diet and like low protein or high protein diets. I think this related to maybe the mRNA or the discussion. Did you already talk about that?
Yeah, we did. Yeah.
Okay.
So we talked about the mRNA, but there's another part where the effects of a low protein diet lingered across subsequent well-fed generations of mice. I think they're talking about rats.
Yeah, and just how like it's I don't have anything really insightful to say about it, but not metabolism as a kind of wholesale like I'm interested in the words like synchronic and diachronic, and like synchronic is things happen at the same time, diachronic is like across time, or across points of time, right? And it's sort of like metabolism is almost a way for there to be a lot of things going on at maybe at the same time, but also across time together. Almost like I always think about the perspective of transferring memories or sort of resonance, even, which is a little bit of a. I feel like I'm stepping into some. I don't know if I want to step into that, dark, deep side of the pool yet with that word, but you have these obstacles to resonance, these obstacles to keeping things in the same place. But metabolism or these other structures, it's almost just like from the perspective of if you're imagine for yourself a small cluster of
molecules traveling at a very, very, very high rate of speed, across a great distance. And like what kind of things would need to happen to have some coherence between those things, and and obviously that's sort of like every moment of that's always that's the norm, and it's sort of like that perspective of what has to kind of be moving in the same direction for things to happen, and talk about the engram in this specific location, and it's interesting to think about kind of, I don't know, what systems do that. So that's sort of one branch of things, and I think you guys have talked about that, so I apologize. And I guess the other thing I'll bring up is let's see. Oh, I knew you guys covered this too, but I forget which animal it was actually, but it was the one where it was there was during its pruning, the pruning of its, I think it was the torpor related discussion. Yeah, and saying how originally they thought when it was coming back, it was like, a fascinating finding is that it seemed to be able to remember very accurately the details and the memories of its kin, right? And it was saying, but it lost a lot of memories, but it can remember its kin. And it said, actually, it might be because of its sense of smell and its like self-referencing through smell being too similar to itself or something like that too. And it kind of doesn't know what I was saying in the sense of, okay, now we have like ways of offloading parts of that process where you can just put it out and pick it back up, integrate as the process picks up. Okay, that's the cue. And I think you guys talked quite a bit about that already in your own, so I apologize for repeating things.
Yeah.
No, you're good.
Yeah, that sounds interesting. So this is the squirrel where they're having to offload things into the environment instead of keep it in their brains because they're losing capacity and then they're gaining it back. And
Right.
if I understand, yeah.
If the same result can happen with something that's like simpler, maybe less demanding, then yeah, it makes sense that would be the route that its development took.
Right. So yeah. So if I go back to I guess Jesse had to leave, or he's going to be back.
Nope, I'm here. I'm just switching back devices.
Okay.
Sorry.
That's fine. Let's go back to the paper. I had a couple more things I wanted to let's look at. So this is talking again about the engram and these RNA experiments, which are the Russians also did some work on this. So the Russians are always come up like doing work that no one's ever heard of because their science community for years was sort of isolated from the rest of the world, so you would not hear about these things. And then they come up like in the history of science, and it's kind of surprising. So the Russians did some experiments with planarians, doing conditioned learning, or sort of operant conditioning, where they would have motor responses, and they'd couple it to responses to a shock paired with a vibratory stimulus. So, this is the sort of conditioned learning and testing that. When animals were transected, retention could be observed in both halves. This is where they had the experiment where they cut the planarians in half. And so this is where they were actually finding the same thing as these studies by Corning and John, where they used RNA, suggesting the possibility that RNA inheritance could be involved. So it's 89 to 91. So there are a bunch of experiments. Let's see, down in 89 to 91, you have, let's see, that's here. So this is in Russian. I don't know. I know that Mike Levin is from Russia originally. So when he was a child, he was living there or something. So he knows Russian. There's going to be Russian references in here. I don't know what that means. This is the paper here, produced by ribonuclease on nervous activity of planaria. So this is back, when we didn't, RNA was working with RNA was in its infancy back then. They were just working with some of these tools and exploring. What's interesting is that there was some interesting work that's been done with C. elegans and transgenerational inheritance, where they would, because in C. elegans you have like a 3-day generation time, so you can actually observe things. And there are experiments you can do with the larval stages, like you can put the worms into starvation immediately upon hatch, and they enter this phase called L1. And there's this L1, or this sort of arrest of development that can occur. So you can put them into developmental arrest at L1, and then you can bring them out of it. So you just put them in a medium. They sit there in L1 they can sit there for, I think, like a week or so. So you can extend their lives by just letting them park in L1 and hibernate. And then when you bring them out, they have some particular adaptations that they can exploit later in their lives. So, there have been a lot of experiments on this. The Biolab, I think at Duke, does a lot of experiments on this, or other groups that do experiments on this. But you can also look at these organisms. You can put them in L1 arrest, and then you can do experimental evolution. So you can look across generations. And what people have found is that there's this transgenerational inheritance, or this epigenetic inheritance. There's certain epigenetic marks that persist across several generations when they're exposed to these kind of conditions. And so they've actually found a similar thing in humans during starvation. There's a data set from, I think it's from the Netherlands or Sweden, where they've looked at grandparents who've experienced starvation, and they look at the grandchildren, and they find that they have certain adaptations that persist. And so they've actually, in C. elegans, they've isolated this to certain epigenetic markers that persist across several generations. So it's not, it's not Mendelian inheritance, it's epigenetic inheritance. It operates by similar rules, but it's a different pathway of inheritance, but it only persists for a couple generations. So there's this whole literature on that's kind of reminiscent of what they're trying to do in planaria. In planaria, of course, they're just working on sort of, what's sort of the seed of memory. But there is this aspect of inheritance and sort of how the mechanism is translated over generations, if possible. But there's other work here. Where's this other paper I was looking at here? Oh, this is James McConnell's forgotten 1960s quest for planarian learning, a biochemical engram, and celebrity. This is a popular article in '96. There's an article, Ginsberg and Jablonka, epigenetic learning in non-neural organisms. So this is another paper on epigenetic learning, and this is in these non-neural organisms. So I'm not familiar with this article. It looks interesting, though. Then there's epigenetic mechanisms in memory formation. There's DNA methylation in memory formation. So there are all these aspects of like learning and memory, molecular bases for memory that are really interesting. Let's see, where were we before? We were in this area talking about planarians. Actually, this talks about the, I guess this is one possible locus for the cellular basis of memory are the planarian neoblasts or stem cells, which could be modified through epigenetic changes induced by learning. When the new brain develops, neoblasts could potentially imprint the central nervous system through self-organization mechanisms. That's actually a series of papers. This is, I guess, some speculation based on these citations. A second possibility is that non-coding RNAs implement inheritance. So this is where they're taking the non-coding RNAs and they're looking at that, and they're suggesting that implements some form of inheritance. I don't really, that's 99 to 100. So there are a number of molecular mechanisms you can propose, but so they're kind of proposing potential solutions to this. Regardless of the specific mechanisms, a complete answer to this question will also require an understanding of the mapping of cognitive content to specific molecular states. So this is where we talked about this several weeks ago in the Saturday meeting, where in the experiments with rats, you had these very specific molecular states or conformational states that the protein had to be in to be active, and then you could pharmacologically block that. So you could actually use something like what they did with the RNase in this paper, or in the examples they gave in this paper, but it's much more targeted because you know that certain pharmacological agents can block certain mechanisms. So we know kind of that maybe there's something like that going on, that there's this we can block learning, we can block retrieval. We know that so the components in memory, if we want to review this, because I think it's important, is that you have acquisition, which is where you acquire information. So if I read a book or a paper, I read it, and I remember some of that stuff. I just kind of, obviously, if you read a book and I ask you what do you learn, you remember stuff. Now, you don't remember every single word. Some people, I think, can, eidetic memories and so forth. But for the most part, let's just say that I, want you to recall certain things, and you'll maybe give me a vague answer or a complete answer or whatever. So you acquire the information and you store it in working memory. So when you're reading, you're kind of like going along. And if I disrupt you in the middle of reading the book, then you have to remember what page you were on, and you may or may not be aware of that. So there's a lot of things that can interfere with working memory, and it's a very finite capacity. So you have this register. People have thought of it as a register where you're just bringing in information in very small amounts and holding it, and then it's moving back and encoding into other types of longer term memory. You do this in your prefrontal lobe. Then that information moves down to this medial temporal lobe, where it gets encoded into a longer term memory. So we want to be able to preserve that encoding. Somehow it gets translated into some sort of larger framework. And then it gets stored in the parietal lobes, where we have longer term memory consolidation. So things get consolidated into long-term memory that way. And I'm referring to the human brain now, but that just or I guess any stock mammalian brain for that matter. So you have three seats of memory, the prefrontal lobes for like short-term memory, the medial temporal lobe for consolidation, and then the parietal lobes for long-term storage. Now, when I probe that memory and I ask you to remember what you did, what you read in the book, and you give me an answer, what memory system is doing is it's retrieving that information. It's going back to the place where it's stored, and we don't know spatially exactly where memory is stored. A lot of people think that it's a network of places, so you have memories. Because you have memories embedded in context, it's stored in a bunch of places simultaneously and has to be retrieved. And that's, you retrieve it from different places, and it's a varying success, that process. Some people also think that in the engram case that it's a specific location for a single memory. And so, that's why we have these experiments. People are trying to say, okay, where is it in the brain? We know that most memories are in the parietal lobe versus the occipital lobe. But other than that, with the medial temporal lobe, if you lesion it, like I said, in the case of HM, you can, remove the ability to remember things. So you sever that system, and effectively you can't remember, you don't have a long-term memory. So you're retrieving things from the medial temporal or from the parietal lobe, it's coming back into play, and you can remember something. And that only holds true really for, declarative memory and maybe episodic memory. Procedural memory is a different thing going on. And then fear conditioning is also another thing going on. So your procedural memory is usually in the cerebellum. And that's a different ball of wax, but it's basically a similar thing where you're retrieving it from a storage. So that's that. I wanted to go through that because I wanted to make sure we're on the same page in terms of how people are thinking about it. Jesse said, would be interested in papers that delve into the divide between memory accessibility and consciousness versus memory storage, or memories being maintained even if not in thought or immediate thought. Yeah, we should do that. We should get together some stuff on that.
I really appreciate you walking through what you have so far too. I
Yeah.
kind of wanted to do that, so it's definitely helpful.
Yeah. So let me kind of finish up where I was going with all this in the paper, because we're getting near the end of the meeting. So this kind of goes through some of these scenarios, and I like this because and then it goes into some memory and aneural systems, which is of course something we're also interested in with respect to some of the other work we've talked about. And so this is interesting here. I think this is Amanda's highlight. Interestingly, neural-like computation, decision making, and memory have been reported well beyond the traditional CNS, including in sperm, amoeba, yeast, and plants. So there's cells that encode memory of things like, different physiological parameters or different growth parameters or things. There are different simple behaviors. These appear to be mediated by well-conserved, ubiquitous mechanisms that appear to be also involved in neural information processing, such as cytoskeleton and electrical networks. So in cytoskeleton, you have, and we talk a lot about this in Devorm actually. There's a line of research that Susan Crawford Young is pursuing on stability in cells and how the cytoskeleton holds a certain shape, and how it has to be held in that way throughout development to maintain the stability of a tissue or something like that. So we actually we don't talk about this aspect of it, but we do actually get into some of that. And then this idea, single somatic cells perform subtraction, addition, low and bandpass filtering, normalization, basically all the operations that computers and signal processing do. So we can do these sorts of things with digital computers. And single somatic cells do this. And I remember actually that this is something that there's a field called cellular decision making. And they talk about this in 168. So let's go down to 168 and see what the reference is. Here it is. This is Christoph Koch here. The role of single neurons in information processing. So this is Nature Neuroscience. This is, I guess, going back to another set of references. There's this cognitive principle challenges clonal selection. That's immunology today. So that may have something to do with the immune system. The cognitive paradigm in the immunological homunculus. That might be kind of interesting. This I.R. Cohen. I don't know who exactly that is, but that would be an interesting reference to look into. Do bone cells behave like a neuronal network? That's another interesting one, and so forth. So there are a lot of references here on that. That would be really interesting. Spatiotemporal dynamics of the electrical network activity in the root apex. So there are actually some papers from plant learning and I know Amanda talked about that, about plant cognition or something like that. So yeah, so that's a great paper. I like that. So we have any other things we want to say before we end?
Yeah, I unfortunately have to go right at 8.
Okay.
But I thought that this paper was interesting because it I was still left asking, like, but where are memories?
Yeah.
But this challenges the idea that, like, you can, I guess, reduce memories to any particular physical thing, or at least we don't know what type of physical thing to look to outside of, like, I mean, we have a general idea, and that's like what a lot of Levin's work is about, which is like bioelectric networks.
Mm.
But then that's mysterious in its own way, um, because it's similarly difficult to, like, localize things. So it this project kind of challenges um, the goal of localizing these functions, I think.
Yeah.
I was also still, it was interesting to me that in the paper there was still this language of encoding and decoding,
Mhm.
because of what I just said, because it seems like these ideas challenge this, like, file cabinet view of memory. Um,
Yeah. Well, I think it's definitely like a thing that, is a challenge to, I mean, what if it doesn't work like that? Because there are other systems apparently that are doing this. But I think the encoding and decoding really is the idea that it's stored somewhere and then it's retrieved. And it doesn't have to be like in a file cabinet, but it has to go through some transformation. It gets stuck somewhere and it comes out. Like, you can't just, you know, just have it moving around continuously, because we know that we can disrupt it. And we know that some of these experiments we can restore it or we can transfer it. So it's got to have some place, and we don't know where. And it may be not one mechanism because we see it in all sorts of systems. But we have these basic rules. There's heavy in learning, there's conditioning, the things you can do, there RNA transfer and things like that that allow you to have a little bit of control over it. So the idea being that there's some encoding and decoding that's going. We don't really know where it is though. It's kind of a still we don't understand that.
Yeah. And I know it's basically time to go. Were you able to record this one, Bradley? Because I wasn't
Yeah,
able to. That's
yeah, I
great.
got it.
oh, thank you. I didn't even think about that.
The last thing, and many have to go, if that's okay. I basically just a plus one in what you said, in the sense that I was thinking the exact same thing. It's like, geez, that paper about coding, and now this, and it's like the thing that comes to my mind is the, like, web development perspective of, if it doesn't exist in three places, it doesn't exist at all. Like, and how, like, if you don't have the backups and you don't have uh, redundancy of memory activation across these different domains, then what is it? And obviously that's depending on the scale and how a neuronal, or what level, complexity, if you will, of the nervous system. It's different, and all that stuff. But it's just the sense of, like, what are the different things that get to work with, and then finally, it's for thing for me to remember is, like, I'm kind of feeling it. What's shifting even after the coding metaphor paper, and this is, like, memory less as, like, a thing and more as an activity, like almost like going back to, yes, riding the bicycle, in that when you're riding your bicycle, there are certain things you can offload from having to worry about. Like, whereas if you're standing straight, you have to hold your posture a certain way. But if you're moving, if your whole stuff is moving in a direction, there are certain things that are much easier to do and less easier to do, or particularly in the aspect of maintaining, because you have other inertias going with it. That's kind of I really want to look into that space, and kind of all of my comments around that today. But maybe in the future we'll talk about that. At any rate, cool stuff, and I think it's been an amazingly successful like tour of, like, the Cybernetics book and Herbert Wiener book and this. So thank you so much, Amanda, for, like, doing this, and I'm looking forward to picking up later. But I think it was a really, really, really great set of discussions. So thank you, everybody, for that.
Yeah, thank you.
Yeah.
All right, see you later.
Be in touch.
Yeah,
Bye.
definitely.